Welcome, AP Biology students, to week 20, and we will discuss the gene regulation. The gene regulation is whether the gene is going to be transcribed and translated or not. And the basic structure for that is the operon. The operon is a regulatory gene sequence that is controlled by one promoter and an operator region. Let's put it into progress. So here we will talk about first the tryptophan operon. This is in bacteria. This is the DNA region of the circular DNA. The tryptophan operon, it contains the promoter region. It contains also a regulatory region, regulatory gene. And this regulatory gene is going to be transcribed into messenger RNA, and the messenger RNA will be translated into the regulatory protein. Now, besides the promoter, we have a operator region. And we have the genes after the operator region. Okay, A, B, and C. These are going to be transcribed to messenger RNA and will be translated into proteins. One more thing that it's missing in here is the RNA polymerase. That will do its job, starting from the promoter, go through the operator region, and transcribe gene A, B, and C into messenger RNA. We will have the ribosomes in here, and they are going to make proteins. Now, we said that the tryptophan operon, so it means like makes tryptophan, an amino acid that is essential for the bacteria. The bacteria needs to make its own, and it takes energy. This is anabolic reaction, requires ATP. The bacteria needs to make the enzymes to be able to make tryptophans for itself. At this point, the regulatory protein would look like this. I'll try to come up with something like this in just using some shapes. So this is the regulatory protein, and this is not active. This is inactive at this point. The regulatory protein, and the tryptophan operon is on. It's going to do its job. This is on at this point. That bacteria will get into a situation when there are tryptophans all around. So the bacteria would think, like, why do I have to waste energy to make A, B, and C messenger RNAs out of those genes and make my own proteins to make tryptophan amino acid for myself when the surroundings contain tryptophans. Here is one tryptophan, here is another one, TRYs, tryptophans, TRYs. And now these are going to combine with the inactive repressor. This is called the repressor. Inactive repressor. So it's going to turn something like this after attached to it, the repressor. Look like that, straightens out. Is a TRY. And this one is right now, it's going to sit at the operator region. So when this repressor combines with the tryptophan, that we call it co-repressor, this co-repressor activates the repressor. So now this is active. And now the active will sit at the operator region. So it's going to turn off the operator as it sits in here. So the RNA, it would look like something like this. So the RNA polymerase will not be able to pass the operator section, will not be able to transcribe the genes into messenger RNA. It saves energy for the cell because it doesn't have to make its own proteins for making tryptophan for itself. So this is a repressible operon. We can repress it with a co-repressor. Normally, this is on, but when we have the co-repressor will activate the repressor, so it combines with the operator region, and it will stop the RNA polymerase action. This is one type of the operon. The other one is the inducible operon. So let's draw this out, the inducible one. So now, again, everything is pretty much the same layout. An operon, here is the regulatory protein, regulatory gene for a protein. We have the transcription, messenger RNA, and we are making a regulatory protein. And I will draw that is like something like this. Okay. We have the promoter region. This is where the RNA polymerase will start the transcription. Polymerase. We have again an operator segment. I use the same shape stuff, but they don't have to be the same. So this is the operator segment. And let's say now we have the genes after this, but it's a little bit different, X, Y, and Z genes. Now, this inducible operon, let's say it's going to transcribe those genes to messenger RNA, that they will make proteins, that these proteins are going to be enzymes that help breaking down the lactose for simple sugar for the bacteria. So lactose, if glucose and lactose is present, the bacteria will use the glucose to make energy for itself. But if there's no glucose, in this case, the bacteria will use the lactose, but it has to be broken down into monomers because lactose is a disaccharide, has to break down into monosaccharides to be able to use it. That's why it needs to make a protein to break down the glucose to monosaccharides. So it's called the lac operon. Lac stands for lactose. Normally, this regulatory protein is active. That means it can combine, goes here to the operator segment and sits here in the operator segment and does not let the RNA polymerase leave the promoter region through the operator region to transcribe X, Y, and Z genes. So the lac operon will not make any messenger RNAs. At this point, it is going to be inactive. This is on off position normally. Now, when we have lactose coming in, so this will be a triangle, that is the lactose. This lactose can sit here or can sit here and makes this regulatory protein leave the operator region. So it's going to be inactive regulatory protein. It will not bind at the operator region, so it will let the RNA polymerase go through the promoter region, operator region, and start to transcribe the X, Y, and Z genes. So it's going to turn on the operator. It will be able to go on. This is inducible operon. The lactose is going to be the inducer to start the RNA polymerase do its transcription. So these are the two basic regulatory mechanisms for prokaryotes that help the prokaryotes to manage whether the transcription and translation would take place or not. So it's kind of their gene regulations. The eukaryotic gene regulation is a little bit more complex, and this will come up later on around in your learning. Have a good one.